right, let's dive into something truly wild today.
Yeah.
We're taking a look at one of the most debated and, frankly, eyebrow-raising scientific ideas from the 20th century.
hmm
The work of Professor C. Louis Kervran and his theory of biological transmutations.
Yeah, this is one of those ideas that right from the get-go, it just flew in the face of, well, conventional physics.
Right.
Kervran proposed that living organisms, like plants or animals, could somehow change one chemical element into another.
Like actual alchemy in biology.
Sort of, yeah, like turning potassium into calcium. And crucially, not through high-energy nuclear reactions, but at the kind of low energy levels you find inside a living cell.
Which, like you said, just sounds impossible from a standard physics viewpoint, doesn't it?
Totally. Conventional science says changing elements requires massive nuclear forces. Energy is just vastly greater than anything happening in biology.
So our mission in this deep dive, using the sources we've got, is to really unpack Kervran's work.
Right.
We'll look at his theory, the experiments he described, the intense scientific reaction he got,
Which was pretty intense, yeah.
and ultimately what this whole controversial journey might teach us about, scientific evidence, the limits of understanding, and maybe the difficulty of questioning established ideas.
It definitely sparked a huge debate, precisely because it hit on such fundamental scientific principles like conservation laws.
And the thing most people remember, the hook, were those famous chicken experiments, right?
Ah, the chickens, yes. Absolutely central to his claims.
We'll definitely get to the chickens. But first, who actually was C. Louis Kervran? Lived 1901 to 1983, French scientist.
Mm.
And what's interesting right off the bat is his background. It wasn't narrow. He had experience in geology, nuclear science, and plant biology.
That mix is key, I think. That multidisciplinary view was absolutely essential to his thinking. He wasn't stuck in just one field's box.
Right.
He was looking across different areas for connections, for patterns, even if those connections seemed, well, unorthodox to specialists.
And that really shaped how he approached science, it seems. He was fundamentally an observer of what he saw as biological anomalies.
Yeah.
Things in living systems that just didn't seem to add up based on the chemistry and physics of the time.
Mm.
And he was willing to challenge things.
Exactly. When his observations didn't fit the mold, he questioned the mold.
Mhm.
And that led him straight to his core theory, biological transmutations.
Okay, so the theory itself.
His really radical proposal was that living things weren't just rearranging atoms chemically. He believed they had some kind of unknown ability to actually change the atomic nucleus,
Wow.
converting one element into another through what he called low-energy nuclear processes happening right there, inside the organism.
Low-energy nuclear processes
Yeah.
in biology. That sounds like a fundamental contradiction. How did he even think that could work?
Well, he tried to explain how, with the proposed mechanism he named frittage.
Frittage.
Yeah, frittage. It was his concept, honestly, pretty vague from a physics perspective for how biological systems, maybe enzymes or something, could somehow facilitate nuclei merging or splitting at normal life temperatures and pressures.
Yeah.
Bypassing the huge energy needed for regular nuclear reactions.
So like a biological catalyst for nuclear change.
That was the idea, more or less. But it lacked a solid physical basis.
And you mentioned earlier the math didn't work either.
Yeah.
That was a big red flag.
A huge one, a fundamental conflict, really. When Kervran wrote down equations for these alleged transmutations, say, potassium turning into calcium,
Yeah.
he wrote them like chemical reactions. But if you check the mass and energy, they didn't balance according to nuclear physics.
Oh.
And violating mass energy conservation, that's basically a non-starter in physics. It drew heavy criticism immediately.
So you've got this extraordinary theory, a fuzzy mechanism, equations that clash with physics. But he claimed it was based on real-world observations.
Yeah.
What did he actually report seeing?
Okay, so this brings us back to those famous chicken experiments, probably his most cited example.
Let's hear it.
As Kervran described it, he had hens living on soil specifically chosen because it was very low in calcium.
Right, calcium deficient.
And predictably, when hens don't get enough calcium, they stopped laying eggs with hard shells.
That makes sense. Eggshells are mostly calcium carbonate. Need calcium for that.
Exactly. But then Kervran reported that when he gave these same hens supplementary mica, which he analyzed as rich in potassium but poor in calcium,
Okay.
or sometimes he used oats, also high in potassium,
Yeah.
the hens apparently started laying hard-shelled eggs again.
Wait, so they weren't getting enough calcium in, but they started getting potassium, and suddenly they could make calcium-rich eggshells.
That was Kervran's hypothesis. He saw this apparent increase in calcium output the eggshells without a corresponding increase in calcium intake.
And concluded.
He concluded transmutation must be happening. The chickens were somehow converting the potassium from the mica or oats into the calcium they needed.
Wow, that's a leap.
It is. Though, interestingly, he wasn't the very first person to notice weird calcium balances in chickens. Way back in 1799, the chemist Louis-Nicolas Vauquelin had noted chickens seemed to excrete more calcium than they ate.
Huh. So there was some historical precedent for the observation, even if not the explanation.
Right. Kervran definitely linked his work to those older, puzzling findings.
That's fascinating.
Yeah.
But he didn't just stick with chickens, did he? He looked at humans, too.
He did. Another key observation he cited involved studying workers in very hot conditions, like desert environments.
Okay.
These workers were naturally consuming a lot of salt sodium chloride to replace electrolytes lost through sweat.
Standard practice.
Right. But Kervran claimed he analyzed their sweat and found it was consistently higher in potassium than in sodium.
So taking in lots of sodium, but sweating out more potassium, his idea was the body was turning sodium into potassium.
That was the connection he proposed, yeah. Another case where the elemental balance seemed off, suggesting an element was appearing or increasing in a way simple input-output couldn't explain.
And plants, too. You mentioned seed germination studies.
Yes, that was another area. He reported observing that as seeds sprouted and began to grow, they seemed to show increases in certain elements.
Elements that weren't there to begin with.
Elements that weren't present or not in sufficient quantities in the original seed itself or in the sterile medium they were growing in. Again, he pointed to transmutation as the explanation for this apparent creation of elements during growth.
Okay, so chickens needing calcium, desert workers sweating potassium, seeds sprouting new elements.
Hmm?
He saw these patterns across different life forms and built his case.
That's right.
I can only imagine the reaction from the wider scientific community when this stuff started getting published.
Oh, the reaction was immediate and overwhelmingly critical. Yeah.
Because it hit those fundamental principles.
Exactly. His claims just directly challenged bedrock physics, elemental conservation, the laws governing nuclear reactions, everything we thought we knew about atoms.
And that energy problem you mentioned earlier, that was the biggest sticking point.
Absolutely. That was the killer criticism from physicists. They did the math. Converting potassium to calcium at the atomic level is a nuclear process.
Right.
And each individual conversion releases a pretty hefty chunk of nuclear energy, around 8 MeV, mega electron volts.
And 8 MeV. What does that mean practically for a chicken laying an egg? Is that a lot?
It's an enormous amount of energy at the scale of biology. If a chicken were transmuting potassium to calcium at the rate needed to make an eggshell every day, the total energy released would be massive.
How massive?
So massive it would, well, vaporize the chicken instantly. Critics famously quipped that Kervran's theory, if true by known physics, would turn his chickens into atomic bombs.
Wow. Okay. That's a pretty graphic illustration of the problem.
It really highlighted the fundamental conflict. If this transmutation was happening, as he described, the energy release predicted by physics was just completely incompatible with life.
Okay, so a massive theoretical hurdle. What about the experiments themselves? Did anyone else try to replicate his findings? Did other people look at chickens making calcium?
People did try. Various groups attempted similar experiments over the years. The results were, shall we say, inconsistent. Crucially, there were very few, if any, independent attempts using really rigorous controls that successfully replicated Kervran's specific claims about elemental increases.
So no solid confirmation from others.
Not really, and a really significant blow came in 1975 from the French Atomic Energy Commission, the CEA.
Okay, the official nuclear agency. What'd they do?
They conducted some very precise experiments. They looked at things like oatseed sprouting, similar to studies Kervran had referenced, but they used much more advanced analytical techniques.
Like what?
Specifically, neutron activation analysis.
Which is?
It's a highly sensitive method. You basically make the sample slightly radioactive and then measure the specific radiation emitted by different elements. It lets you detect incredibly tiny amounts, much more precise than the chemical methods Kervran likely had access to.
Got it. So the CEA used these super precise methods on the oat seeds. What did they find? Did the elements change?
Their findings did not support Kervran. They reported no significant increase in calcium, sodium, or manganese as the seeds sprouted.
No transmutation.
No sign of it. They did observe a small decrease in potassium, which you'd expect the seed is using its stored potassium to grow.
Right, normal biology.
Exactly. But absolutely no evidence of new elements being created. This was seen as a major experimental refutation of Kervran's claims.
So the rigorous analysis by the CEA didn't back him up at all. But you mentioned before some interest kind of lingered.
Yeah, it's interesting. Despite the mainstream rejection and the CEA results, a few researchers did continue to explore what they saw as anomalous elemental stuff in biology.
Really? Like who?
For example, there was work by Komaki in Japan in the 70s and 80s looking at microorganisms, bacteria, molds, yeasts like Aspergillus niger, or Saccharomyces cerevisiae, common baker's yeast.
And what did Kamaki report?
They reported observing some unusual shifts in elements during the growth of these microbes. It suggests that for a small fringe, Kervran's questions, or maybe just the underlying odd observations, kept the possibility alive even without wide acceptance.
Okay, let's swing back to the theory side for a second. Kervran had this fridge idea. Did he ever flesh that out more? How exactly did he think biology could trigger nuclear reactions?
His ideas on the mechanism remain quite speculative, honestly. He sort of hypothesized that enzymes or maybe complex biological structures could act like catalysts by somehow guiding protons or neutrons between atomic nuclei or lowering the energy barrier. But again, it was never presented as a detailed, testable mechanism that was consistent with physics.
So it didn't really convince physicists?
No, it didn't gain traction because it couldn't provide a convincing physical explanation.
And that fundamental energy problem always remained the biggest hurdle, right? He just couldn't explain how biology, this low-energy environment, could possibly provide the massive energy needed for nuclear changes.
Precisely. The proposed mechanisms, like fridge, never managed to bridge that gap in a way the scientific community found plausible.
So, if the mainstream wasn't buying transmutation, how did they explain Kervran's observations? The chickens, the sweat, the seeds. What were the alternative, more conventional explanations?
Well, the alternatives were generally much less dramatic and crucially consistent with established science.
Like what?
The most obvious one was simply undetected contamination.
The classic experimental problem.
Exactly. Could Kervran's calcium-deficient soil have actually contained tiny trace amounts of calcium? Could the mica or oats have had more calcium than he measured?
Plausible.
Over time, a chicken's biology is very good at concentrating elements it needs. Even tiny amounts in the feed could potentially accumulate and account for the calcium found in the eggshells. Eliminating all trace contamination is incredibly hard.
Especially back then.
Right, which leads to the second alternative. Simple errors in the analytical measurements themselves.
These measurements might have been off.
Measuring trace elements accurately is tricky, even today, but especially with older techniques. Critics argued Kervran's methods might just not have been precise enough. What looked like an increase might have been within the margin of error or just analytical noise.
Okay, contamination, measurement error. What else? Could the organisms themselves just be shuffling elements around?
Absolutely. That's the third category, known biological mechanisms. Organisms have sophisticated ways of handling elements.
Like concentrating them.
Exactly. Biological concentration is well known. Plants and animals can selectively absorb certain elements from their environment, even if they're present in low concentrations, and store them.
Or get rid of others.
Yes, selective absorption or selective excretion. Maybe the desert workers weren't making potassium. Maybe their bodies were just becoming really efficient at conserving potassium under heat stress while readily excreting the excess sodium they were consuming. That could look like a shift in the sweat composition without any actual transmutation.
So these alternatives, contamination, measurement error, biological concentration, selective handling, they all sound much more, well, ordinary.
They are, and much more consistent with existing biology and chemistry. That's why they were and remain these strongly favored explanations within the mainstream scientific community.
Extraordinary claims require extraordinary evidence.
And Kervran's evidence, when scrutinized for these potential mundane explanations, just wasn't seen as extraordinary or robust enough to overturn fundamental physics.
So, taking all this perspective, how does Kervran's work connect, if at all, to modern science today? Does it resonate anywhere?
Well, his specific theory of biological transmutation isn't accepted, but his work sort of touches on themes that are still areas of active, sometimes fringe research.
That's it.
You could draw a faint parallel to fields like cold fusion or LNR, low-energy nuclear reactions.
Oh, yeah, that's also controversial.
Highly controversial, yes. But the underlying theme is similar. Exploring the possibility of nuclear reactions happening at much lower energies than conventional physics predicts. Kervran was doing that, but in a biological context.
Interesting parallel. What about within biology itself?
Modern biology does still encounter anomalies that aren't always easily explained. Sometimes researchers find unusual isotopic ratios in biological samples.
Isotopic ratios.
Yeah, variations in the flavor of an element based on its neutron count. Sometimes these ratios in living things are a bit different than you'd expect from just the environment, and explaining the exact biological processes behind that fractionation can be complex.
So, still some puzzles.
Definitely. And there's ongoing research into biological energy pathways, how life manages energy so efficiently, sometimes in ways that seem counterintuitive.
And things like quantum biology.
Right. That's a fascinating emerging field. It looks at whether quantum mechanical effects, the weird rules of the subatomic world, might play real roles in biological processes like photosynthesis or how enzymes work.
Did that support transmutation?
No, not directly at all. But it does open the door to thinking that biology might harness physics in ways that are more subtle and complex than we previously assumed. It suggests there might be layers of physical phenomena at work in life we're only just beginning to appreciate.
Which is super interesting.
Yeah.
But let's bring it back to Kervran's specific work. Thinking about his methods, how do we assess them now with hindsight? What were the strengths and weaknesses?
Well, on the plus side, you have to give him credit for being a systematic observer.
Okay.
He conducted studies over long periods. He used multiple subjects, chickens, humans, plants. He tried to quantify things with measurements, and he documented his observations detailedly in his books. Those are good scientific impulses.
Definitely sounds like a scientific approach.
It does. But the limitations, as we've discussed, were critical and ultimately, from a mainstream view, fatal to his claims.
The controls.
Yes, major issues with experimental controls, especially regarding potential contamination. It's just so hard to rule that out completely without modern clean room techniques and ultra-pure materials.
And the measurement accuracy.
The analytical precision available then was limited compared to today. Could he reliably detect the tiny changes he was claiming? It's questionable.
And the big one, replication.
A huge limitation. The lack of consistent, independent replication by other labs using rigorous methods. Science relies on reproducibility, and his results just weren't reliably reproduced.
You also mentioned statistics earlier.
Right. His work generally lacked the kind of rigorous statistical analysis we expect today to show that an observed effect is real and not just random chance or variation.
So despite the systematic observation, the methodology had significant gaps by modern standards, especially for such a revolutionary claim.
Exactly. The evidence just didn't meet the required burden of proof.
What about the impact of his work, though? Even if the theory wasn't accepted, did it influence scientific thinking at all?
I think it did, indirectly. It represented a bold challenge to the established paradigm.
Pushing boundaries.
Yeah, he was questioning the clean line between chemistry, rearranging elements, and physics, changing elements, suggesting biology could somehow blur that line. While his answer wasn't accepted, asking the question, poking at that boundary, can itself be valuable?
And his broad background.
That highlighted the potential value of interdisciplinary thinking, bringing ideas together from physics, chemistry, biology. That approach is much more common now in tackling complex problems, like in systems biology.
And it serves as a lesson in methodology, right?
Absolutely. The Kervran story is a classic case study in the scientific method. It underscores why rigorous controls are crucial, why independent replication is non-negotiable, and why new theories, especially radical ones, need to either align with established fundamental laws or provide truly overwhelming evidence to overturn them.
The scientific community's reaction, while critical, was essentially upholding those standards.
Pretty much, yes. It was a defense of the established process of validation.
So, looking at Kervran today, is there any real contemporary relevance beyond that historical lesson? Does his specific idea pop up anywhere?
You sometimes see echoes of it, but mostly in niche areas, often outside mainstream, validated science.
Like where?
Some proponents of certain alternative agriculture methods, or maybe some fringe nutrition ideas, might occasionally reference concepts similar to biological transmutation, but typically without rigorous scientific backing. There's also ongoing, though often controversial, research into unconventional energy processes in biological systems that might distantly relate.
But generally, his core theory isn't part of accepted science.
No. Kervran's specific transmutation theory hasn't found a place in modern biology or physics textbook knowledge.
So if we do that critical assessment now, scientific validity today,
From the standpoint of conventional evidence-based science, Kervran's biological transmutation theory lacks sufficient, robust, replicated experimental evidence. And crucially, it faces that fundamental conflict with the laws of physics, particularly energy conservation. It just doesn't meet the high bar for such an extraordinary claim.
But the historical significance is different.
Yes, I think so. Historically, his work is significant not because he was right, but because he was an example of a scientist willing to grapple with observations that didn't fit, willing to question deeply held assumptions.
That act of questioning is important.
It is. That willingness to investigate anomalies while still demanding rigorous evidence is a vital part of how science, ideally, moves forward. Even failed challenges can teach us something.
So the key lessons for science today are about that balance, rigor, and openness.
Exactly. You absolutely need the rigor, the controls, the replication, the theoretical consistency. That's essential for reliable knowledge. But Kervran's story is also a nudge, perhaps, to keep an open mind towards puzzling data, towards phenomena that seem wrong, while maintaining that critical skepticism and demanding strong proof.
Okay, so let's try to wrap this deep dive up. We've explored Professor C. Louis Kervran and his really extraordinary, definitely controversial theory of biological transmutations.
hmm The idea that living things, like those famous chickens, could somehow convert one element into another, like potassium into calcium, at low biological energies.
And we've seen that while he was clearly a careful observer, his specific claims just haven't really held up under sustained scientific scrutiny.
Right. They ran into major problems with fundamental physics, especially the huge energy requirements for any nuclear change. And crucially, they lacked that robust, independent replication under tightly controlled conditions.
So his work ends up being historically significant, maybe not for proving transmutation, but more as a story about challenging assumptions and the importance of investigating anomalies rigorously.
Yeah, it really makes you think about how science vets claims and how knowledge progresses or sometimes hits dead ends.
And it's fascinating that even though Kervran's subpific theory isn't accepted, modern science is still uncovering surprising complexity in biology.
Absolutely. From subtle isotopic effects to potential quantum influences, it reminds us there's likely still a lot we don't fully understand about the intricate processes of life.
So Kervran's legacy, maybe, is serving as this compelling case study, a reminder of both why we need rigorous scientific methods, but also the value of keeping a genuinely curious eye on those observations that just don't quite seem to fit the current picture.
Well put.
All right, here's a final thought to leave you with. Given just how incredibly complex living systems are and how much we still don't fully understand about, say, biological energy transfer, or even the potential role of quantum mechanics in life, could there possibly be natural processes happening inside organisms that are even stranger, perhaps even touching on elemental transformations in ways our current models don't fully capture than we currently imagine?